thank you for the kind and accurate introduction. And thank you all for coming. It has been really a pleasure to be here today. Such a welcoming environment. I think it's the Midwestern hospitality. It's really different from Boston. But cool. So I just want to give you a little story about what these things are. These are particles. As you can see, they are not natural. They are perfectly spherical. They are artificial particles that are made of food for bacteria. And all of the bright green things there are bacteria that have been stained. And I love this picture because it just shows the diversity of colony morphologies that you can get there. Even without knowing anything about species, this looks really intriguing. So I'll tell you more about these things from a different perspective later. So I want to start here, which is not what I'm doing now, but what I did before. And I just have 2 or 3 slides about this to put things in perspective of why I'm doing what I'm doing. I think it is important to explain that. So I used to be a computer scientist at some point. I wasn't very good at that. But I used to be more into genomics and statistical genomics in particular, and studying evolution of microbes and taking advantage of this great platform that Martin Post has at MIT with all these closely related organisms. And so after all that work, we published a review paper that was kind of my second PhD thesis after the postdoc with Martin. And the claim that we wanted to make here is that ecological interactions play a significant role in shaping the diversity of genomes. And in this illustration, what you have, so you cannot see it very well, but the tree is a species tree. So all the branches are, let's say, things that are part of the same species. But they can be subdivided in populations where, for example, one population lives in this part of the lake, this other population in the other part of the lake, but they're the same species. And if you look at the genes in these organisms, you will find things that they all have in common. So that will be the first column. That you cannot see my thing there, but the first column. So that's what makes a species a species. I mean, they all have the same genes. And there are other things that are specific to the population. The fact that this one lives in that part, in that habitat, and maybe have to deal with different nutrients or something, so it has its own set of genes. So if you don't know that, and you just look at a species, you may call those genes in the second column flexible genes. And I hope you're familiar with this terminology of core genes and flexible genes. So core genes are shared by all members of the species, flexible only by some. But this is not really flexible it has ecological meaning. It's something that's specific for a habitat. And so we thought it would be useful to distinguish that from those things that I would call really flexible, which would be the other 2 columns, where things are always or in general at intermediate frequencies or low frequencies in local, well-defined populations. And so it turns out that there are different mechanisms, genetic mechanisms by which you can get that, but at least we had 2 pieces of evidence that supported the idea that ecological interactions were the drivers of that intermediate frequency. Hello. And so there was 2 papers that I published back then, and in both cases, so the first one shows that there are clusters of genes dedicated to antibiotic production that are only in 40% of a local population, but everybody in the population is resistant. And in the second one with siderophores, only a small fraction of the population has genes to produce siderophores, which are molecules that are secreted to the environment and allow organisms to acquire iron. But everybody in the population can take those things. So they are cheaters, if you're familiar with cheater-cooperator sort of narrative. And so in particular, what we saw was that you had these operons. You can basically look at, it's not so much about the genome, it's just operons. There is an operon on the left that has transporter genes in green and the biosynthesis genes for the siderophore in orange or red. And there are other members of the population that have only the transporter genes. But everything else around in the genome is the same. So it's just this little piece of DNA that is coming in and out to make something a producer of this public good. So which is, I think is remarkable. And just to finish this introduction, nowadays we're still trying to follow this up. And we have found very similar patterns with other type of secreted proteins. For example, alginate liases. These are the enzymes that degrade alginate, an important biopolymer in the ocean, antibiotic production. You see the same type of thing. Xylose metabolism, you also see this differentiation at the level of operants. So that's the prelude I was referring to. And the statements I want to make from this is, it seems that ecological interactions are significant enough that they have an impact on genomic diversity. That's one thing I feel confident saying. And the other is that they must have some impact on, I suppose, on community dynamics and function. But really what that impact is, finding that out is difficult. And I want to highlight that all these things I just told you in these 3 slides, this is what I would call the bottom-up approach, where we start with a genome or something in culture, try to make sense of why it's different from this other genome, and try to put that back into an ecological story that is higher up in the level of abstraction. And so, right, so remember that. I was talking to some people today about the experience of starting a lab, and I do remember it very clearly that moment where I literally put my feet on the table and I thought, okay, I can do whatever I want. But what is it? And one of the things I identified was just what I just told you, this challenge of going top down and trying to find out how interactions impact dynamics and function, and not just, you know, staying with the level of genes or one organism and so on. And so this is a really difficult challenge I have been learning. It's more difficult than I expected. And one of the difficulties is that it's very difficult to learn ecology in the lab. It's difficult in the environment because, as you know, it's hard to go beyond descriptions. And you bring things in the lab and you can be quantitative and mechanistic, but you know, what's happening in the environment versus the lab is a big problem. But I did feel that it's more difficult than just sequencing and doing sequencing and statistics. You need more than that. So you need, at least for my taste, some type of more detailed control of things. So I tried to find compromises for that. So you will see what I mean by that. One of the difficulties of doing these things, of learning ecology for microbes, is that the problem of scales. So this is one of our main contentions, that people have traditionally been doing things at the really large scale, you know, up there, and for good reasons, because, for example, you care about biochemical cycles, so it's better to measure the scale of the whole lake or many liters of water or grams of soil. And for other reasons, people are interested in the very small scale, the micro scale, single cells. But I would claim there is a sweet spot in between where the actual community ecology takes place for microbes. So where, for example, things aggregate, they interact, and those interactions impact the way they aggregate, or the dynamics, and so on. And so I wanted to look at those things. That's one of the sort of founding ideas of the way we do things. And I would say a prime example of those sweet spots, mesoscale structures, are particles of organic matter. Okay, so what you have here in these pictures on the left, these are plant fibers in the rumen of a cow. On the right, this is a poor dead copepod being eaten up by bacteria. So in both cases, at a certain level of abstraction, this is just a complex material, a pretty big thing, bigger than the head of bacteria, if you can say that. And the bacteria need to aggregate around the thing, attach, secrete enzymes to degrade it. Yeah, and so that's the kind of, to me at least, I can sort of wrap my mind around this. Okay, this is a really interesting ecological process that I can relate to community ecology happening at so the microscale tens to hundreds of microns. That's the kind of thing that I like to understand better, and see how interactions with I suppose they're likely to occur in this case, since things are so close to each other, how they impact the function and dynamics. And so to try to start converging with this lengthy introduction, so we have projects that are starting on the animal-associated communities, in particular things that degrade plant fibers in cows. That's why I put that there. But we have been working for most of the time I've had my lab on marine particles. So I'm gonna for the rest of the talk just gonna talk about that. And in the case of the ocean, so marine particles, I'm sure you guys know what this is. It's a huge bloom of algae. So I'm just going to tell you what's the story of where these particles come from, but many of you already know this. And the main idea is that you can see the ocean as a biodigester, so not so different from a gut at a certain level of abstraction, of course. But so this is basically, from that perspective, the planet eating tons of salad. Okay, so it's a huge amount of carbon that is being introduced into the ocean. And, but because things die for many different reasons, and because algae tend to excrete polysaccharides and all these things happen, carbon is essentially released, either in the form of already hard materials like the shells of diatoms or you know, membranes and stuff like that, or the copepods that grow, that eat them, they also die, and a lot of hard shells come out. Or also polysaccharides tend to aggregate because they're sticky. And eventually, you get all these particles forming, right? And many of these particles tend to sink because they're heavy. And so, but remarkably, most of them are eaten up in the top 500 meters of this digester. And so what people think happens is that there's a bunch of bacteria that have adaptations to attack the particles. And these people call them opportunitrophs. I kind of like that because they're floating around waiting for this particle to come their way, and then they attack. And to attack, they basically have the ability to attach to particles. They have carbohydrate binding proteins, for example. They can secrete enzymes, which is a remarkable thing to think about in the ocean. You're floating in this environment, and you're just pooping out enzymes that will diffuse away, and some of them you're hoping will hydrolyze the particle and will give you food back. And so that obviously releases a lot of nutrients, also by byproducts of metabolism. Could be acetate, could be amino acids. And that sort of triggers the assembly of a community. This is, I think I like to say, it unlocks the niche for many other things to go and co-assemble. Eventually, supposedly, these particles are eaten up, and then bacteria need to restart the cycle, go find new particles. So that's the logic of this whole thing. And of course, this whole thing happens at the landscape scale. So there are many particles, so the full game is how these things move between particles and whatnot. So that's what I hope we understand in the next few years. We're slowly making progress in this direction, but this is really our goal, to figure this out. And the approach, we're starting to get there, is the following. So remember these particles I showed you at the beginning? So this was actually this thing here, which is, it looks like the moon, but it's just, in that case, it's a chitin particle. So the whole thing is made of chitin. But it could be different biopolymers, as you will see. But these are, in general, gels. So it's not legit chitin. It's a gel that has, it's chitosan that has been reacetylated to become chemically equivalent to chitin, but it's a gel. And so you can make any shape with that. So it's a little bead. The dark thing there is a magnetic core, which allows you to pull them back from the environment. And it's very small, 50 microns. And so that's the first thing. What we do is we make microscale enrichments in the environmental microbiology sense, and enrichment. You modify the environment to allow certain things to grow. So sure enough, bacteria like chitin, some of them, and they will attach and grow and make these colonies on the surface. And then we can, of course, do a lot of sequencing. So we have all this omics data. And that tells us something about the dynamics of colonization, for example. And then we also love to do culturing. So we culture bacteria from the particles. And in general, we are quite successful in recovering most of the taxa that attach, because the particles themselves are an enrichment. So it's a pre-selection from all the things that are in the ocean. Only those things that are grown particles, they tend to be more easy to isolate. So we get many of the taxa that we see here in the culture-independent data, we can get in culture, which is great, because then we can map those we can map the reads, let's say, to the isolate, so we can see what the dynamics was for each isolate. We can do phenotyping, see what they eat. Do they secrete the enzymes? Do they swim? Things like that. And we can relate that to the assembly process. And ultimately, we can do what I call synthetic ecology, which is basically reassembling communities and try to find the logic of how these things work. So that's the approach. That's what we're trying to do. And so I'm going to finally tell you about the first story, which is something that was published. So I'll try to be brief, and then I'll move to things that are at different stages, but all kind of approaching publication, or at least submission, which is quite different. So, right, so what we did was we took about a liter of seawater, and then we put there about 1 million particles. And this is very dilute, actually, because the particles are very small. And we calibrated this to avoid collisions and aggregation of particles. And we have them rotating overhead to create the flow. And then at different points in time, we collect, because the particles are magnetic, otherwise you cannot see anything. We collect about 1,000 particles per time point, and we did 16S and full metagenomic sequencing to get the taxa and the genes. And we also do the culturing that I told you before. And this is the main result, I guess, that when you look at all these taxa, here is time, and these are the taxa ordered by time of arrival, is that you get this order succession where something is coming in, it peaks. So a peak would be red, and then it disappears, which is black. And then something comes in, grows, and in the meantime, something is coming in, growing, and so on. It keeps going and on. This wave of invasion and dispersal, apparently. This is, by the way, total abundance, as far as we can tell from, we did the qPCR thing. So it's not just relative abundance, which means that these things are really living the particle. And the other interesting thing is that I can tell you from this is that we had 3 replicate bottles. So I guess biological replicates from the same place of the environment. We sample the water, put them in different bottles, repeat the same thing. And any taxon that we pick here, you will find basically the same type of dynamics on the different bottles, which is somewhat remarkable because there are about 100 things there. And so if I see one taxon appearing at 70 hours, I can tell you also appear 70 hours in the other replicates. So what controls this program is the question, I guess, and it's not fully clear. But we have some ideas. So this is the data I just showed you, is the sequencing data. And this is a slice of this matrix, so just a few rows of the matrix, for those things for which we had cultured OTUs. So we had isolates that we could map to the culture-independent data, right? So that means that for these things, we can now do these phenotypic assays and try to find what's different between those that come early and those that come late. And the result is I think, really interesting. So obviously, these are the early ones, these are the late ones, and these are the things that we tested in culture with the isolates. And there is a remarkable distinction between early and late. So all the early strains, or isolates, species, whatever you want to call them, they can grow on chitin, they can degrade chitin. They secrete the enzyme, yeah, so they excrete that thing. They can grow really well on the monomers of chitin. They can grow on the dimers, chitobios, and they are motile. You know, so we do these assays where you can stab soft agar, and then whoosh, they swim really fast. And to first approximation, that's completely the opposite for the late colonizers, which is surprising. I mean, there are some exceptions. There are things that seem to be able to grow on chitin but not secrete the enzymes. Maybe they keep it bound to the membrane. There are a few things that can grow very well on the monomers, maybe like a classical cheater that would take advantage of the monomer without producing enzymes. But for the most part, they seem to be inert. So I'll tell you what we think happens to those. But by now, so we didn't have this when we published this paper, but by now we have all these genomes, so we can look at the differences and also remarkable differences in the genomes. This is what we call the primary degraders, and this I will call secondary consumers. And so when we look at the genomes of primary degraders, we see things like this. This is just a little fragment of the genome of one of these primary degraders. This chitinase, and it keeps going. I'm very proud of this animation. Chitin. And this thing you have here is something called MSHA locus, which has been shown experimentally to enable attachment of vibrios to chitin. but it's not, this is not a Vibrio, so it's a more general thing. And so there's experimental evidence that if you knock this out, things don't attach very well. It's a type of pili. And so all this in one cluster in the genome, there were, I think, 8 chitinases there. In total, this organism has 20 chitinases, which kind of blows my mind. This organism is a pretty small genome for bacteria. It's like 3 million base pairs, and it's just basically devoted to destruct chitin. That's what it does. And it's, not surprisingly, an early colonizer, as I told you. So we can map, obviously, like I told you, the dynamics. But what happens with the late colonizers, they don't have as many chitinases. And actually, I should say they do have some chitinases, which is surprising because they don't ground chitin. But they still happen, 1 or 2. But by and large, they don't have nothing like this. And so what we saw, however, is that when we culture them together, I will explain the figure. When we culture them together, you can see that the primary degraders basically enable the growth of the secondary consumers. And this is shown here, where on this axis you have the primary degraders, and this is the control. This is the monoculture of the secondary consumer. There are 2 in this case. And the bars is the fold growth from basically the abundance of the secondary consumer at time 0. So I should say the abundance of secondary consumer at time final divided by secondary consumer at time 0, and compared to what happens in monoculture. In monoculture, there is no growth, basically, as I told you, but in co-culture, they can really grow significantly, like a thousand-fold increase. So this is basically, and it's quite general, right? It doesn't matter too much who is there, they just are facilitated. And so what we think happens here is some general type of cross-feeding. And I'll tell you more about this. We don't know exactly whether the chemicals that are being cross-fed, what is the food, but it is most likely it's not a derivative of chitin because they are not good at eating that. So it has to be some other byproduct of metabolism. And this could be many, many things, amino acids, acetates, stuff like that. And just to wrap this up, this is something I really like, which in general, I'm not a big fan of measuring diversity although I know it's like the main thing in ecology, but I tend to find it not very useful. But in this case, I was very surprised. It was my bias, I think, because I was very surprised that this was quite informative, I felt. So you start at time 0, diversity increases because things are colonized. That makes sense. And then diversity decreases significantly, but then at the same time, and this is for metagenomes, and this is the only piece of data I show you from metagenomes, like when you can look at all genes, the frequency of chitinases, the genes that degrade chitin, increases very nicely and smoothly and reaches a maximum at the same point where diversity is minimal, which is basically, I mean, it's not the best word, but it's a good word. It's a kind of selection, but it's not in the sense of evolution, but in the sense of species sorting. So diversity decreases, and the things that survive this bottleneck are the things that have a chitinases. That's why you see that. Right, so that's a pattern, which I think is really nice to see it there. And then diversity goes back up, which is amazing. And then the frequency of chitinases decreases. So that's the second invasion of the secondary consumers that don't eat chitin. So I like this because you can see it without looking at species or anything. You can just see the main pattern of the assembly. Okay, so that was that. Oh, I always forget to click through the things, but it's basically what I just told you. So now moving to things that are still in the, yeah?
Does the population that's not attached to the chitin change?
Oh, yeah, good question. We did check that. They do change, yes. You put things in a bottle and they change, but the change is uncorrelated with what.
Matter if you have the chitin beads or not?
We did not do an experiment where we put the chitin beads. Sorry, we did not put the chitin beads and just look at that. But from the same bottles, we sequence just the water, and the changes are uncorrelated. Yeah, it's a good question. And so now moving into things that are to be submitted, I guess. So we repeated this thing with different biopolymers, and this is something to be completely fair. The students and Manoshi, who at the time was a postdoc, they came up with this idea, and I wasn't really sure it was a good idea, but it turned out to be a good idea. And so because what it allows you to ask really interesting questions that I will try to explain. And so, for example, first question is, I mean, what type of dynamics do you get? Are the successions specific to chitin, or is it something more general? And well, that answer is easy. So you get the same type of dynamics in all these substrates. Okay? But now, what this allows you to do is to ask, well, I mean, this row here is a specific taxon. It's not the same one as here. They're just ordered differently, right? So it could be completely different species in each substrate, or it could be a small set of species that overlap. And if so, where do they overlap? At the beginning, at the end? I mean, it allows you to define how narrow or broad is the range of each taxon, and at what point during the assembly of the community this broad or narrow range taxa appear. And so, to do that, I hope this was clear, but I'm going to try to explain it a bit better. So you can look at any taxa. I'm saying taxa. You can think species, OTU, whatever. And you can say, okay, so this one here, for example, this is an illustration, obviously, but it grows in agarose. It doesn't grow in alginate, it doesn't grow in carrageenan, and it grows in chitin, and so on. And so you can try to come up with an index to say, okay, this is somewhere in between. It's not completely a specialist, but it's not really broad range. Whereas something could grow on everything, in that case, it would be a broad range, and so on. And so what we do is we sort of get rid of time by getting an average abundance, and we use a geometric mean for that. And then we calculate an entropy, which is, it complicates the explanation, but it's basically that's our index for how narrow or broad range things are. And then when we calculate these things, these numbers for each species, we plot a histogram, and surprisingly, there are basically two clusters of things. So on this side, on the left, you have the things with very low entropy. Those basically appear only on one type of substrate. Yeah, that would be a low entropy. And so a proper name would be to say these are narrow range taxa, but I call them specialists, but bear in mind that this is only based on the distribution at the moment. And on the other end, you have the generalist, and again, this is a misnomer, but it's a more broad range taxa. But so I'll keep using the word generalist on the other slides. But anyway, the point is that they segregate in these two groups. And so that's interesting that we can just from the sequence, from the patterns of occurrence, we can distinguish these things in an unsupervised way. And this actually shows it quite nicely because these are the same type of heat maps I showed you before, but they have been put next to each other. So, but this is basically for agarose. These are the dynamics, and then right after comes alginate, but time begins again at 0 and 0 and 0. So there are basically 5 heat maps put next to each other, right? I hope that's clear. And so you see all these things basically have the same dynamics and are really abundant on all particles. And these are the narrow range or the specialists, right? And so here's our entropy measure that I was describing. So it shows that it does capture the pattern. But actually you should see it there by eye more easily. So you can see that there are these groups of taxa that appear specifically on each of the carbohydrates. And I don't have all this data on these slides, but some of them we have in culture again. We can look at the genomes, and not surprisingly, it's like the chitin case that they have tons of enzymes to destroy that biopolymer, but not other things. So I think those things are real specialists. It's okay. So these are the dynamics of the so-called specialists. So you can see, I mean, they increase primarily in one of the substrates. But what's interesting is that the broad range taxa or generalists have very similar dynamics on all the substrates. So it's not just being abundant on all the things, they also do about the same. For example, this weird guy increases and then decreases in all substrates, and so on. There are other examples. So this guy here keeps increasing on all substrates. So it seems to be really substrate independent what they do. I don't have a slide about this, but there are other really interesting aspects of this whole study. So we made combinations of, for example, particles that are mixed of agarose and other substrates. For example, agarose, alginate, agarose, carrageenan, and so on. And what we find is that these mixed polymers basically recruit a linear combination. The community is a linear combination of the community of the individual biopolymers. So basically, you get recruitment of the specialists for each of the biopolymers, which I think is interesting. But anyway, so the last thing I want to say about this is that so we can ask, well, so if all these generalists, or I should call them again broad range taxa, appear at the beginning of the succession and the specialists appear at the end, what you would have effectively is that the dynamics of the communities are diverging towards their specialists. And of course, the opposite would mean that the dynamics are converging. And so we can ask that question. And so what we do is that we add them all up, all these generalists. These are all the broad range taxa, the generalists. You add them all up, what you see is that they tend to increase in abundance from 0.2% to 20%. So basically, the communities are converging. They start, now we have better visualizations of this, but they basically start with their specialists that again maybe sort of unlock the niche, and then come the broad range generalists that invade the community. And the last important fact I should mention about this is that these generalists, which again we have in culture so we can test what they do, they don't degrade all substrates. So they are really not generalists in that sense, right? So they are just broad range. What we really think happens is that they are generalist cross feeders, so to speak. And to me, this the way I interpret this thing is basically the community dynamics is sort of enslaved by the dynamics of metabolism, or by the structure of metabolic networks, I should say rather, where you have a lot of diversity on the periphery of the network. You can plug in a lot of different things. You can produce a lot of different enzymes to degrade different substrates, but eventually everything turns into simple small sugars that funnel down to glycolysis, the EEB pathway, or the pentose phosphate. So it's really funneling down to simple things. The same with amino acids, everything can. So to me, this means that cross-feeding is likely to be this process where you start with generalist things that are specialized and end up developing into a community of generalist cross-feeders that could appear anywhere, regardless of the initial substrate. So that's kind of what I think we are learning from this. All right, so I'm going to switch to the last. I have this intermezzo. I forgot. So I have 15 minutes more, right? Yeah, so I'll be fine. Don't worry. I see faces. So, but the next few things should wake you up. So this is an intermezzo. It's something that is there, and I just want to show you because I think it's a really cool question that we're also working on. And the question is, why do things actually leave the particle? Many people ask that. And the answer is we don't know. But we have something that we have found in the lab that I think is the first indication of that this actually is possible. So yeah. So why do we have this sort of decrease from once the primary degraders have colonized, they conquer the particle? Why do they leave? Actually, the why should be on the next phase actually. And okay, so pay attention to this because it will happen very fast, but the movie will loop. But pay attention to the time scale. So you would see there is a red thing there. That's a cell, and more cells will come, but it's all super fast. So this is a strain of Vibrio coriolyticus. It's not something that we isolated. It was from Asaf Bardi's lab, but it's a really good chitin degrader, and this is a chitin particle. Okay, so you see it colonizes really fast, and then it detaches. It happened there, about 10, 12 hours, I think. And then it reattaches again. Right, so can you see that? So it will start soon, and then whoop, and then detaches. Okay, and so when we quantify that, and it's something that's really reproducible and very quantitative, you see these things on the red line. It increases on the surface and then abruptly declines, and it's as fast as the colonization. It's like, and then it moves to the medium, where it becomes motile and so on. So the exact signal for this is something we are investigating. What is it? My guess, if I may speculate, I think this is the fact that as degradation initiates, monomers are released, and these monomers are a signal that triggers motility, and they prefer to swim around rather than stay attached. So I think it may relate to a foraging strategy. If there are enough of these monomers, you actually want to leave and not stay attached to one particle. But it's the first indication we have that actually this detachment is real and it's something we can measure and we can possibly control in the lab. Okay. So now the last part of my talk is related to what we can define as function on these communities. And the cool thing, and this is more movies that should wake you up, is the cool thing is that we can do time lapses of the particles, and surprisingly, or not so surprisingly, I don't know, but for us it was cool to see the particles are eaten up in the lab. So, and we can measure that and be quantitative about it. And so there you have the same particles. On the left, you have the same Vibrio correlyticus that I showed you before. This is a different color. And this is just phase on the right. And so this one is a really good degrader. So it happens pretty fast. About 30 hours or 40, you see the particles collapse. Okay, as these things grow on the particle, then the things collapse. Yeah, and our other degraders that we collected from our real particles, I mean from our incubations, most of them are not as good. So for example, this one takes about 120 hours, 150 hours, something like that. So what you're seeing here is the volume of the particle over time normalized by the initial volume, that's why it's 1. And you see nothing really happens for most of the time, and suddenly there is a little bit of swelling. It's something that you expect from hydrogels. As they get degraded, water comes in the matrix, and then there is an abrupt collapse. And so this type of dynamics allows you to, and this is the growth of the organism over time, which was surprisingly hard to measure because things are attached. So we ended up using DNA as a proxy. But you have basically exponential growth during this whole process. And only at a certain point you get the collapse of the particle. And, but the simple dynamics allow you to measure a half-life of a particle. So you basically collapse all these dynamics into one number, which is how long it took to reach half the size, the volume rather. And then a different degrader with the same initial number of cells will have, in this case, about half the half-life. So a significant difference. And there is another one that can go even lower. So really large differences in the hydrolytic power per isolate. And these things we think relate to the number of enzymes they have in the genome and so on, but it's a more complicated story. All right, so the first question is, I indicated sort of superficially that that comparison between the green and the red curve was with the same number of cells, but actually you should think that the number of cells that you start with this experiment should matter significantly for the outcome for this half-life, right? So if you start with 1 cell, if you start with 1,000,000 cells, that half-life can change. So we thought, okay, we should look at this and also try to distinguish between two competing hypotheses. That one is that because the organisms excrete enzymes, the idea, at least conceptually, should be that there is a cooperative growth. So if you're producing enzymes and releasing some monomers, I can benefit from that, and you can benefit from my monomers, and we're all sort of cooperating. And this should be cooperative growth, which means a rate that accelerates with the number of cells. And the alternative hypothesis is that there is no such thing, it is purely competitive, and you have exponential growth. And the prediction from, if you just make simple models of this, is that the time, this half-life will scale with the initial number of cells in different ways. So for example, if it is exponential growth, the scaling is with the logarithm of the initial number of cells, and if it is cooperative growth, the scaling is with the inverse of the initial number of cells. And then, so obviously we did experiments. This is a nice one. For example, here we're just diluting the initial number of cells, and you see it takes longer and longer the more diluted the initial condition was. Yeah? And this is in logarithm scale. And so sure enough, what you see is that the half-lives depend linearly with the logarithm of the number of cells, which indicates that there is no cooperativity. So it was just good to check that. No cooperativity. This is what the model would predict, a linear scaling if there's no cooperativity. If there's cooperativity, the time it takes to reach this half-life would increase disproportionately as you dilute more and more and more, because things cannot find each other to cooperate. So that's simply what's happening. And okay, so I'll move to the final punchline here, which is how this effect, the degradation of particles by primary degraders, how is it affected when we include the cross-feeders that I was telling you from the beginning, the secondary consumers, I call them, right? So basically make the simplest possible community with a primary degrader, a secondary consumer. What do you get? And so this is the same data I showed before, the primary degrader at a fixed number of cells. We chose that number, 125 per microliter. And now we add a little bit of this secondary consumer. It happened to be a strain of Maribacter, it's a type of flavor bacteria, if you care about those things. And it takes a little bit longer. If you add a little bit more, it takes a little bit longer, and so on, and so on, and until actually you can basically choke the primary degrader. The whole thing gets inhibited. And right, and this is something that we see only when we have particles. If we do this in liquid, there is no inhibition at all. Actually, this secondary consumer, it can barely grow in liquid on the monomers. So we do the test, we try to see chemical inhibition, nothing like that. It seems to be a phenomenon that is related to growth in particles. And so so this, what I showed you before was sort of tuning the concentration of the secondary consumer, but we can also tune the primary degrader. And what we see, this is the data I just showed you before, but what we see, and I hope this is clear, it's not completely clear here, but that is the effect we get is dependent on the ratio between primary degrader and secondary consumer. It's not so much about the actual numbers. And that's one thing, because, yeah, that's one thing. For example, this was the data I showed you. To inhibit, we needed to have this many cells. But to inhibit at a lower concentration of primary degrader, we needed less cells. And the second statement is that when we do the same with a better degrader, we need many more cells of the secondary consumer to get the same effect. So the susceptibility of the primary degraders to this effect depends on their hydrolytic power. So it seems to be some type of race between how fast you can degrade before the secondary consumer sort of invades and damages your ability to degrade. And the last thing I want to show about this is that, almost the last thing, is that these degraders are actually effectively the secondary consumers are effectively parasites. And what this means is that they get a benefit from the primary degrader, but they hurt back. So it's like a plus-minus interaction. So these are number of cells of primary degrader during degradation. At this point, you have degradation. If you add the secondary consumer, the solid line is the primary degrader. The dashed line is a total community. So the difference is the secondary consumer. And it just takes longer. And if you add more secondary consumer, it takes longer. And if you add too much, nothing happens. And so basically, it's a kind of suicide of the community if the load of secondary consumers is too large. But the statement here is that the primary degraders are not growing as much. Their rate and yield is diminished, and that's why basically you have inhibition. Right, these things I already told you. Yes. Okay, and the last thing is sort of speculative, but I just wanted to connect the things I showed you before with the dispersal. So what you're seeing here is this data is what I showed you before, that movie where I showed you there is attachment and then decrease and dispersal. It's the same data. And actually this recolonization reaches really high levels of cells attached, and then this decline is the collapse of the particle. Yeah, I was just showing you the early stages of this before. And this movie here is the same type of thing, but when we add the secondary consumer. And I'm sure you cannot see this very well, but it's taking really long. So it's going all the way here. And actually it's taking really long. But there is an initial explosion of cells, colonization, and then they leave the particle, but they just don't come back. That's what happens. And I'm sorry, this data is not really, it's all normalized to 1, that's why it's so high. But you have, again, an increase, then a decrease, and they just don't come back. And so with this, I want to say, basically, the mechanisms of apparent inhibition seem to be related to the behavior of the of the cells around the particle. Either the gradients change, or there is occlusion of the resources by colonization of the other organisms. So it's not a chemical interaction per se, it's more related to their behavior, which is interesting but complicated. Okay, so you survived. So summary, I think this is actually a bunch of rants rather than summary, but so I told you at the beginning we're going to do things top down rather than bottom up, and when we do things bottom up, we can find all types of interactions. When we do things top down, cross-feeding, and by this I mean trophic interactions, so exchange of carbon-containing substances, seems to be the interaction that dominates dynamics. So that's one thing. Well, so there's a certain role for metabolism. That's the cross-feeding. There is behavior, whether you attach or detach, you swim or not, and physical interactions. And all these things come together to determine what's happening. And cross-feeders behave as parasites. There is a negative feedback occurring on the degraders, and that has potentially significant effects. Like, for example, particle degradation is completely blocked, or at least the increase is significant in half-life. Yeah. And then the other thing I didn't mention really explicitly, but all these interactions that we find are not specific to one strain. We can reproduce them with different strains from the same functional group, like primary degrader, secondary consumer. The actual numbers change, but the phenomenon is the same, which I think is important. Right, so I like to thank primarily those people involved, but this is my group now, and funding sources, and thank you for your patience. Yeah, yeah.
So then I'm wondering, is that trait of this tile, very quick growth and association, is that very universally present in primary colonizers?
Yeah.
And second part of the question, is this a trait that's also lost in some of the primary consumers that you see in long-term culture or last?
Yeah, that's good questions. I don't have the answers, but because the problem is that to actually see that, you need fluorescent label strings. So we are working on the labeling. We have had some success, but we haven't checked that yet. But there is indication that at least for Vibrios is universal. As if the universe was Vibrios, it would be universal. But yeah, it's well conserved across Vibrios. And so I don't know for the other type of things that we have as primary degraders are not so different, Alteromonas and other gammas. So maybe. But I'm not sure. And it could also be chitin specific, which is another thing that I'll be more worried, if worried is a good word, but that'll be more my guess that it's chitin specific. And I don't know what happens. We haven't done evolution in the lab or anything like that. Mm-hmm.
I think following up on Vince's question, have you tried, is it being driven by protein that the status, or if you were to do the primary degraders and the particles in the absence of the secondary consumers, would they still?
Yeah, the movie I showed first was only primary degrader.
Only primary.
Yeah.
So they do it in the absence.
Yeah. It was different is that when you have primary, secondary consumer, they don't come back.
Okay.
Yeah. Yes. Yeah, good question. I mean, I think I've been looking for these numbers. It's really hard to measure, to be fair. And I don't do the measurements, so. But then the numbers that I've seen out there is like the residence time of particles in the mixed layer tends to be about 100 to 200 days, that kind of order. And so, which is a good point because these are really easy. This is an easy problem. It's a gel. It should be pretty easy. And it takes them about a week in this media that is where there's plenty of everything except carbon. So I'm not sure what happens. How can real chitin, which is so difficult, be actually degraded? I don't understand this. Yeah. Yeah, that's a good question. Um, I'm not sure. So there are two things. So I mean, basically, we don't know. We're just going to start doing metabolomics to try to find what's the interaction there. But it is true there are so many different taxa. I don't think there are as many different substrates, possibly, I think. And so my answer would be that I don't think the system is at equilibrium. And that's why so if you let them run and keep passing them, you probably reduce the number of species significantly, because you'll get the best degrader of whatever, histidine or something that is secreted, I don't know. Yeah? Which one? Yeah.